All right, welcome to Chapter 7, Part 1. In this chapter, we're going to take metabolism a little step further, and we're going to look at how organisms generate ATP from their food. In the first half, we're going to focus on aerobic cellular respiration, and then in the second half, we'll look at anaerobic cellular respiration and fermentation. So think back on the ATP cycle. Remember that all cells need ATP. ATP is the energy currency of the cell. It's going to allow us to take the energy from nutrients, convert it to ATP, or make ATP, and then use that ATP to shuttle that energy to all the other processes in the cell that require energy. So they're coupled. Remember, coupled reactions. Where in this case, the catabolism of our nutrients fuels the anabolism of ATP, and the catabolism of ATP fuels the anabolism of the endergonic or anabolic reactions in the cell. And so ATP is continuously recycled, and that's really important to remember. We're constantly making ATP. And cells, so no cell can survive without ATP. I mean, there are other energy molecules in cells. GTP sometimes, PEP, phospholipyruvate, but ATP is the big one, and all cells are going to utilize it. It powers almost every activity in cells. Ultimately, in most living organisms, energy is coming from the sun. And then photosynthesis, which we'll look at in Chapter 8, photosynthesis is going to take that sunlight and combine inorganic carbon dioxide and water to form glucose and release free oxygen. And what we're going to do is the opposite of that today, or the reverse reaction. We're going to look at taking glucose and oxygen and then running it through aerobic cellular respiration to produce carbon dioxide and water and release ATP as a product. And of course, we're losing heat energy. Remember your second law of thermodynamics. A little bit of heat energy is lost at each step. Ultimately, we can break our aerobic cellular, there's three different respirations a cell can do. Aerobic respiration, which we'll cover today, anaerobic respiration, and fermentation, which we'll look at in the next lecture. Now, you got to be careful when you see the word respiration that you don't confuse cellular respiration with breathing. Breathing, as a multicellular organism, you're exchanging gases, and this is occurring at the organismal level of the hierarchy of organization. We're bringing in oxygen, and we're releasing carbon dioxide. And when you bring in the oxygen, your respiratory system and your circulatory system are going to work together to move that oxygen through your body to the cells. And it's at the cells that we're going to use that oxygen to harvest energy from food molecules, and in turn produce carbon dioxide. It's going to be released from the cell, released into the bloodstream, taken back to the lungs, and then released in your exhale. So you can't have cellular respiration without breathing as a multicellular organism, because we need to get oxygen and we need to be able to get rid of CO2. But breathing is not cellular respiration, and we're not making any ATP when we breathe. So how is cellular respiration related to breathing? What you could do is you could pause the video and you could take a few moments and answer this question. And if I'm going to answer this question for you, or I'm answering it for myself, cellular respiration occurs at the cellular level, breathing occurs at the organismal level, but I can't have cellular respiration without oxygen. And as a multicellular organism, I need breathing to bring in oxygen. And I can't let carbon dioxide accumulate. I got to get rid of it, and breathing is going to allow me to do that. Now, before we go too much further, we need to talk about oxidation reduction reactions, or when they're combined, we call them redox reactions. And the real key to redox reactions, the key to metabolism in general, is understanding the movement of electrons and how energy is transferred when those electrons move. An oxidation is when you either gain an oxygen or you lose electrons. A reduction A reduction is when you gain those electrons or you lose an oxygen. So here in this example, we're looking at the losing and gaining of electrons. And compound A is losing 2 electrons to become oxidized, and at the same time, compound B is gaining those 2 electrons and becoming reduced. Because these two things are happening at the same time, concurrently, we say that these two reactions are coupled. And in our cells, we're going to see that there are special molecules called coenzymes that are involved in this process that are going to be they're going to accept the electrons, and we're going to see they accept protons too at the same time. But when these type of reactions occur, we combine the names to call them redox reactions, and you're going to see these extensively throughout the rest of this chapter and in the next chapter on photosynthesis, because that's a redox process too. The coenzymes we're most interested in these reactions. First is NAD, nicotinamide adenine dinucleotide. And you don't need to know nicotinamide adenine dinucleotide. NAD is good enough. And NFAD, which is flavin-adenine dinucleotide, or FAD. Both of these, this is their oxidized form. And both of these will become reduced by gaining 2 electrons and 2 protons to become NADH plus H plus and FADH2 respectively. There's another coenzyme that does a similar process in photosynthesis in the next chapter. That's NADP, nicotinamide adenine dinucleotide phosphate, which would become NADPH plus H plus. And then there's another coenzyme A, usually just abbreviated CoA, that isn't involved in redox reactions. Instead, we're going to see it's an escort molecule. It binds to what we'll see as a 2-carbon acetyl group, and then helps to transport it into a pathway called the Krebs cycle or the citric acid cycle, depending on which textbook you're reading. If I look at the overview of aerobic cellular respiration, I'm going to take a single 6-carbon glucose molecule, C6H12O6. I'm going to need 6 diatomic oxygen molecules, and ultimately I'm going to produce 6 CO2 molecules, 6 molecules of water, and I'm going to generate about 36 ATPs of energy in eukaryotic cells. There are 4 different pathways that are going to help me get to this. Glycolysis, oxidation of pyruvate, citric acid cycle, and oxidative phosphorylation. And it takes a lot of pathways to get there, because we can't release all this energy at one time. Okay, we can't release all this energy at one time. It's kind of like if you have a bonfire, and well, here's a better example. You got gas in your car right now, right? And you can't check the level of gas by opening the gas tank and lighting a match, because if you did, all that gas would explode and it would blow up the car and blow up you, and it'd be a big mess. But when you burn gas to make the car move, you're igniting tiny amounts of the gas at a time, exploding it to make your pistons go up and down, turn the crankshaft, turn the drive shaft, and make the car move. And so by releasing just a little bit of energy at a time, you're controlling the use of that energy source, and you're not releasing so much energy that you would damage the car. Well, the same thing is true in cells. If we released all the energy in a glucose molecule in one reaction, too much energy would be released in one shot, and it would damage cells. But by releasing it in a gradual, slow, stepwise process, now we can generate that ATP safely. And that's what happens in the 4 parts of aerobic cellular respiration. The first part is glycolysis. Now, glycolysis is actually a 10-step pathway. But I don't need you to focus on every individual step and what's happening. I want you to focus on the overview of what's happening. And the overview of what's happening is a single 6-carbon glucose, and the carbons are represented by the little gray spheres. 6 carbons, there's a glucose. Ultimately, we're going to convert that into 2 molecules of pyruvate or pyruvic acid. In the process, we're going to have to invest 2 ATP, but we're going to get 4. We're going to double our money, so to speak. And we're also going to take 2 of our coenzyme NAD, and we're going to reduce that coenzyme to NADH plus H plus. So we're going to get 2 of these. Okay. Now, nearly every organism performs glycolysis. It occurs in the cytoplasm of all organisms, and it's anaerobic. It doesn't use any oxygen. But you don't get a lot of ATP. 2 doubling it may seem like a lot, but we're going to see that's not very much later on. We can think of glycolysis as occurring in 2 parts. The first is an energy investment part. The second is an energy payoff. And when I think of energy investment, I remember when I was a kid and people talked about making money, a lot of times they'd say, you got to spend money to make money. And so suppose that somebody in class had a really great idea of a product we could manufacture and then market and sell to the public and make a lot of money for all of us. Well, we don't have raw materials and we don't have equipment. We don't have a facility to work in. We don't have the help. We're going to have to advertise. We're going to have to ship. It's going to be a long time. We're going to spend a lot of money before we ever get any money back on our product. And the same thing is true in glycolysis. Early on, we're going to have to kick some energy into it to jumpstart the process. But then later, the energy is going to come back in such a way that it pays off our initial investment and we get a little extra to boot. Okay, so the energy investment phase is in the first 4 reactions. And we're going to take 2 ATP molecules, and we're going to start with our glucose, our 6-carbon glucose. ATP is going to come along, and it's going to catabolize and give off a phosphate, which is going to attach to the glucose. And then we're going to isomerize this molecule and convert it from glucose to fructose, which is a subtle change. And then we're going to take another phosphate and donate another another ATP, donate another phosphate, and form this 6-carbon molecule of phosphate at each end. And then we're going to split that in half. And if we're going to make ATP, enzymes are going to make the 2 halves exactly the same, a molecule called G3P, or glyceraldehyde 3-phosphate. Now, you don't need to know these intermediates, and you don't need to know what's happening at each individual step. You need to know that we're investing 2 ATP, we're taking our 6-carbon glucose, and we're splitting it into 2 3-carbon molecules, each with a phosphate on the end. Now, if I'm going to make ATP, I'm going to, at this point, I'm going to start modifying these 2 3-carbon molecules. First thing I'm going to do is I'm going to oxidize this 3-carbon molecule, and in the process remove 2 electrons and 2 protons to reduce an NAD plus to an NADH plus H plus. Okay, and this is happening twice because I had 2 3-carbon G3Ps. When this happens, it creates a binding site for a phosphate to attach. And so the next step is now to let this phosphate be donated to ADP, and enzymes are going to put them together to generate ATP. So at this point right now, not only have I got my original investment of 2 ATP back, but I've also got these 2 NADH plus H pluses. These have potential energy that I can harvest later. I'm going to then isomerize the molecule, move things around. I'm going to then kick off water, dehydrate it, and then I'm going to use the second phosphate to phosphorylate another ADP to make 2 more ATPs. I'm left with my 3-carbon pyruvate, and now here's my 2 ATP that are profit. So I invested 2, I got 4 back. That makes a net of 2 ATP. Now there's still potential energy in the pyruvates, and there's still potential energy in the NADH plus H plus. But I'm done with glycolysis at this point. So again, my summary occurs in the cytoplasm. It's an anaerobic process. I have to invest 2 ATP, but I'm going to get back 4 for a net of 2. I'm going to use redox reactions to oxidize the G3Ps. That's going to reduce 2 NAD pluses to NADH plus H plus. And then ultimately my 6-carbon glucose is now 2 3-carbon pyruvic acids. And I still have potential energy here in the NADH plus H plus and in the pyruvate. So why do the reactions of glycolysis occur step by step instead of all at once? Give you a second to think about that. You can pause the video, take all the time you want. Well, remember that enzymes have an active site that recognizes a substrate, and then they're going to make some kind of a chemical bond change in the substrate to create a product. Enzymes can't make that many changes all at once. They're limited on how much change they can make. And so the changes, and you saw too that there were a lot of different things that were happening. Sometimes we were adding a phosphate, sometimes we were taking a phosphate away, sometimes we were changing the molecule slightly by moving things around. There was once when we lost water, other times when we were losing electrons and protons. And so an enzyme can only do one of those things. It can't do multiple things. And so that's why we have to have multiple steps, and it's a pathway instead of a single reaction. Now, to get energy from the pyruvate and the NADH plus H plus in a eukaryotic cell, we've got to move those into the mitochondria. This is what the mitochondria does. This is why the mitochondria is such an important organelle. But it isn't cheap to do this. Those 2 ATP that we got from glycolysis, we're going to spend them to move the pyruvate and NADH plus into the very middle, the very center of the mitochondria. This area is called the matrix. This is the outer membrane. This is the inner membrane that's highly folded, highly convoluted. The space between the two membranes is the intermembrane space, and the very middle is called the matrix. And that's where I've got to get the pyruvate and the NADH plus H plus to start harvesting that potential energy. And it's going to be worth it. We're going to get a whole lot of ATPs, way more than the 2 we spent to get them in there. Now, the first thing that has to happen, or the second event, is pyruvate oxidation. We've got to modify the pyruvate so that it's ready to go into the Krebs or the citric acid cycle, 2 different names for the same thing. The pyruvate has 3 carbons, and the first thing we're going to do is we're going to oxidize it, and we're going to kick off 2 protons and 2 electrons. And so another NAD is going to be reduced to NADH plus H plus. And this is happening. Here's the outer membrane. Here's the inner membrane. This is the inner membrane space. Here's the matrix. And the matrix is where this is occurring. So we kick off 2 protons, 2 electrons. We're also going to decarboxylate. When you decarboxylate, you lose a CO2. This CO2 is going to diffuse out of the mitochondria, out of the cell, into our bloodstream, and then be released in our lungs as exhale. And now we've got this 2-carbon acetyl group. The last thing we're going to do is attach it to a molecule of coenzyme A to form what's called acetyl-CoA. And the reason we need to attach it to the CoA is because when we get to the next pathway, the Krebs cycle, the first enzyme of the Krebs cycle can't recognize the acetyl group unless it's attached to CoA. But the CoA won't go with it into the Krebs cycle. It just gets it there. And so that's why we call it an escort molecule. So again, if I want to summarize pyruvate oxidation, this is happening in the matrix of the mitochondria. I have to lose a CO2. I have to reduce an NAD to NADH plus H plus. There's potential energy there for later. I've got to attach to a molecule of coenzyme A. And because, remember, we formed 2 pyruvates, all of this is doubled. We're getting 2 acetyl-CoAs. We're getting 2 NADH plus H pluses. We're releasing 2 CO2s from each initial molecule of glucose. The next step of aerobic cellular respiration is the Krebs cycle. This is also known as the citric acid cycle. And each time we go around this cycle, we're only going to get 1 ATP. So not a lot of ATP generated here. But there's going to be a bunch of redox reactions where we're going to harvest a lot of protons and electrons, 3 to NADs to form 3 NADH plus H pluses, and 1 to an FAD to form FADH2. There's potential energy in all of these. And we're going to kick off 2 CO2s. So 2 carbons come in, we kick off the 2 as CO2. Those go out of the cell, out of your exhale. We get 1 ATP. We're going to reduce and form 3 NADH plus H pluses, 1 FADH2. And remember, because we had 2 pyruvates, we got 2 acetyl-CoAs, we're going to get 2 cycles out of the citric acid cycle. Okay? So for each glucose molecule, here's our acetyl-CoA coming in. But see how CoA kicks off? CoA doesn't come into the cycle. We start with a 4-carbon molecule, oxaloacetate or oxaloacetic acid. Again, you don't need to know the names of these intermediates. This is going to attach to the 2 carbons, and now I've got a 6-carbon citric acid or citrate. This is why this is called the citric acid cycle. Then we're going to decarboxylate. We're going to reduce an NAD, and now we've got a 5-carbon molecule. We're going to do those two things again, but this time we're going to form an ATP before we then take our 4-carbon succinic acid and we kick off 2 protons, 2 electrons to reduce an FAD. Without those 2 protons and 2 electrons, it's a different 4-carbon molecule, malic acid or malate. And then we reduce that, or we oxidize that to reduce another NAD, and that modifies this to the molecule we started with. This is why it's a cycle, because the molecule we start with is the molecule we end with. Ultimately, going around twice, we're going to release 4 molecules of CO2. We're going to produce 2 ATPs. We're going to reduce 6 NADs to NADH plus H+. We're going to reduce 2 FADs to FADH2. And there's still potential energy in all of these electron carriers, these reduced electron carriers. At this point, the 6 carbons that came in from our initial glucose molecule have all been lost as CO2. 2 were lost during the oxidation of pyruvic acid. The other 4 just were lost here. So if I stopped right now, I've only gotten 4 ATP. 2 from glycolysis, 2 from the Krebs. And I spent 2 of those to get into the mitochondria, so I really only have 2 ATP. I've released 6 CO2s, 2 from the oxidation of pyruvate, 4 from the Krebs cycle. So all the carbon that were part of the glucose molecule have now been lost as CO2. Ultimately, I've reduced 10 NADs to NADH plus H+. 2 from glycolysis, 2 from the oxidation of pyruvate, and 6 from the Krebs. I also have 2 molecules of FADH2 that I got by reducing 2 FADs. Okay, so at this point, I've completely oxidized my glucose and it's gone. And I've removed as many electrons and protons as I can, or I could have, from my initial glucose. But there's still a lot of potential energy in those electrons and protons, and that's what I want to try to recover next. The process that does this is called oxidative phosphorylation. And this is where we're going to produce most of the ATP that we can generate during aerobic cellular respiration. And this is really the only pathway where oxygen is required. And we're going to see why it's required in a little bit. Now, where this occurs is really 3 places in the mitochondria. It's going to use the inner membrane and then both spaces on either side of it, the intermembrane space and the matrix. And it's 2 different pathways that make this up. The first is a pathway called the electron transport chain, and the second is a pathway called chemiosmosis. So let's talk about the electron transport chain first. All right, electron transport chain. What does it mean by electron transport chain? Well, suppose you were in a room with 7 other people, and you were last in line, and a person came in. Or one of the people had brought a cheesecake, a chocolate cherry cheesecake, a good one. And they take their slice, and once they have their slice, they don't want the cheesecake quite as much as the next person. And so they pass it on to the next person, and they take a slice. And once they have their slice, they don't want quite as much as the next person, so they pass it on. And each time the cheesecake is getting passed, it's losing a slice. It's getting smaller. Until finally it comes to you. And all 6 pieces of the cheesecake are gone, and you get the platter that has cheesecake leavings on it, those little bits and pieces of cheesecake shrapnel. You're the terminal cheesecake acceptor. You're getting the cheesecake in its lowest form of energy. And that's kind of what happens in an electron transport chain. In an electron transport chain, in the inner membrane of the mitochondria, there is a series of proteins that are all connected to each other that have electron carriers within this complex. And these electron carriers will accept electrons that are being carried in by the NAD or the FAD, and they're going to shuttle these electrons to each other in a series of redox reactions. When you have the electrons reduced, when you give them up, you become oxidized, and whoever gets them becomes reduced. And each time they pass off the electrons are losing a little bit of their energy, kind of like the cheesecake lost energy every time it was passed off, until finally the cheesecake is at its lowest energy state, and it's going to be picked up by oxygen because oxygen is our terminal electron acceptor. Okay? And so what we're doing is we're harvesting the energy from the electrons. And what we're going to do with that is we're going to use it to fuel these proteins where they act as proton pumps. Remember, H plus is the symbol for a proton. And we're pushing, the protons were carried by the NADH plus H plus and FADH2. They're carrying these protons across, they're pushing them into the inner membrane space. Now, as more and more protons accumulate in here, they all have the same charge, and they don't want to be there. They don't want to all be on one side of the membrane, so they're pushing against each other, and this creates a force, and we can take advantage of that in the next part of this process. In the meantime, our oxygen that picked up the extra electrons at their lowest energy level it gains a couple of protons to become water, and water is a waste product from this process. Those protons that accumulated, again, they're repelling each other. They're pushing against each other up here. They don't want to stay here. But the only way they can get back to the matrix is through a fourth protein, which is an enzyme called ATP synthase. And when the protons diffuse through ATP synthase, it causes this enzyme to spin. And the mechanical energy of its spinning puts a phosphate on ADP to make ATP. For every NADH plus H plus, we're going to get 3 ATPs. For every FADH2, we get 2. Okay, so there were 10 NADH plus H pluses. 10 times 3 is 30. There were 2 FADH2s. 2 times 2 is 4. I can get 34 molecules of ATP from chemiosmosis by using the energy provided by the electron transport chain. And that's what makes this such an efficient way to generate ATP in the cell. So if I look again at the summary, my electron transport chain is harvesting the energy of the electrons through a series of redox reactions and using that energy to push protons across the membrane into the inner membrane space. They don't want to stay there. The only place they can come back is through ATP synthase, causes it to spin, puts a phosphate on ADP to make ATP. Meanwhile, some of these protons bind with the oxygen that picked up the extra electrons to form water as a waste product. This water is going to diffuse out of the mitochondria in the cytoplasm, and if there's too much in the cytoplasm, out of the cytoplasm into the cell. Ultimately, we generate 34 ATP from this process. Okay. Now I know this is a lot of information, but you just got to spend some time on it, and the more times you work through it, the easier it's going to be. Okay, so let's do a discussion question. Where is ATP synthase located in eukaryotic cells? So go ahead and pause the video, take a second to think about it. Well, ATP synthase was right after the electron transport chain, embedded in the inner mitochondrial membrane. So D is the correct answer. Another discussion question. What does oxygen do in the electron transport chain? Well, oxygen was our terminal electron acceptor. It's picking up the electrons when they're at their lowest energy level, and then it's going to pick up a couple protons and become water as a waste product from the process. So if we put it all together, okay, glucose, we got our 2 ATP, but remember we got to spend those 2 ATP to get into the mitochondria, so we're really not gaining anything there. But what we're gaining is the pyruvic acid, which has energy, and the NADH plus H plus. When we oxidize pyruvate, we get acetyl-CoA, which has potential energy, and we get some more NADH plus H plus. When we go through the citric acid or Krebs cycle, we get 2 ATP because we go around twice. We get a whole bunch of electrons and protons, which we're going to put all those together to work through oxidative phosphorylation. And between the electron transport chain and chemiosmosis, that's the real advantage. That's where all the energy is. And that's why they say, the electrons and the protons are the main source of energy in cellular respiration, because we can recover them here. And now our total is about 36 ATP for our initial molecule of glucose. And that's a pretty good day. So at the end of this part, here's the key terms that you should familiarize yourself with and be prepared to define on the exam. And these are the key concepts that you should use to test your knowledge. If you can answer these from memory, you're ready for the test.